So I'm Loic Martin. I work at the CEA in the institute which names Hibitex, and at the CIMO Pro, which is the service of engineering of molecular proteins. So I've developed a mini CD4 which is a mimetic of the CD4 protein. So by transferring in a small scaffold the elements of the interaction of the CD4 with the GP120, so we obtained a mini CD4 protein, and we optimized. So it took about maybe almost 10 years of optimization, and we finally obtained a peptide which has sub-nanomolars for almost all the clades of GP120 at just show at the exception of clade AE, and now we know the reason why. So the peptide show good neutralization against a lot of clades, and we also show in recently published paper in collaboration with other partners of the CHAARM consortium that it was quite potent to inhibit the cervical and rectal tissue model explant, and we also perform a macaque challenge, and we show that the macaques which received the vaginal application of the mini CD4 1 hour before the challenge, high dose of challenge, because we have challenged with a high dose which present about 10-fold the animal doses which infect 50% of the animals, and we show that in this group which received the mini CD4 gel application in egg gel, we protect 5 out of 6 animals. Only 1 animal presented infection, and we have tried to understand why this animal was infected, but it doesn't seem to be a form of resistant virus, because we sequenced the virus and we didn't show the difference of resistant mutation. So the next step could be the human trial, but to be honest, in comparison with non-nucleoside retrotranscriptase inhibitors and NHI inhibitors, our mini CD4 remains quite expensive to produce. So we are currently trying to reduce the cost of production of such a peptide by 2 ways. We try to improve still the potency of the mini CD4. So we are currently trying to optimize with further optimization of the peptide. So we have already a better peptide which we named M48U12. So maybe we could reduce the concentration of the peptide into the gel. And we also try to perform recombinant expression of such a peptide. For that we tried expression in E. coli, but also mammalian cells, and also Pichia pastoris expression. In our hands, for now, unfortunately we don't have a high level of expression, but we could have quite good expression when we fuse with a partner fusion. So now we are currently trying to express a fusion protein between our mini CD4 and a VHH we have just recently obtained, which is a kind of mini CCR5 VHH. It targets the CCR5 binding site of JP120. So we are currently trying to perform such a fusion protein, and we hope, because we know that VHH is quite well expressed in, for example, in E. coli or in Pichia pastoris. So we hope that we could obtain good fusion protein, high level of expression, and maybe we can hope to have a much more potent compound or fusion protein, because we will target both the CD4 binding site and the CCR5 binding site. Yeah, we will try. So we have still 1 year to perform such construct. We have already performed the construct in the plasmid construct, but now we have to express, to purify, and to test in cells oocytes.